on behalf of our Young Managers Forum, though I'm not young, but of course, on behalf of our Young Managers Forum and Lucknow Management Association, I extend you all a very warm welcome. And of course, Dr. Ramesh Upadhyay, who is joining us from Sweden. As I'm told, Dr. Ramesh Upadhyay is joining at the moment from Sweden. It's my privilege to put few words about him, to introduce him for this very interesting and knowledge sharing session this afternoon. Dr. Upadhyay is a serial entrepreneur and a distinguished scientist specializing in infectious diseases like HIV, tuberculosis, and other neglected tropical diseases. Currently, he is the Chief Executive Officer at Laxmi Life Sciences, Hyderabad, and also Chief Scientific Officer at Home Oncology at United States of America. Though I can see he has a pretty long profile. It will take enough time to read out, but I'll share only a few things which are important. Dr. Upadhyay is also a scientist at Division of Nephrology at Harvard Medical School, Boston. He's also distinguished visiting professor, Plasma Bioscience Research Center at Seoul, Korea. Another important feature which I find here, he is keen to develop new business across India with primary focus in Uttar Pradesh, as we are speaking from Uttar Pradesh and its state capital, Lucknow. The key areas being healthcare and innovative technology, to name a few. So here are the few words about Dr. Upadhyay. As he rightly just stated that he'll be sharing just few slides. And as we have shared in our communication mail also, members who are joining us, who have joined us, can raise any kind of question to Dr. Upadhyay, either in chat box or ultimately through direct posing their question to Dr. Upadhyay, and he'll be happy to respond to those questions. So with all these words, I welcome you all once again, and in particular, Dr. Ramesh Upadhyay, who is joining us from Sweden at the moment. So with these words, I hand it over to you, Dr. Ramesh Upadhyay. Please.
Great. Thank you very much, Rajiv ji, for making that nice introduction. And especially to Gaurav ji, who has been instrumental actually to put up a seminar like this. So what I always believe, that based on this particular session, that there is due to this pandemic, there are a lot of questions on the mind of people. And at the same time, there is all kind of information that is floating around. So what we thought that, okay, if we will look into this particular information related to SARS-CoV and Omicron, and try to understand and gather this knowledge that will make us free from the fear. And at the same time, knowledge always helps actually to, first of all, to dispel the fever and then prepare ourselves and fight it well. So with this, I will try to dive into a couple of those slides. And then, as we rightly said, we are going to have that wonderful session on the question answers as well. So going to the next slide, the idea that I have that, okay, what kind of a matter I that need to cover during the presentation. So first of all, SARS-CoV biology, life cycle, diagnostic and nomenclature. Comparison with other RNA viruses, how unique it is. Features of Omicron, pathogenicity of Omicron, and immune response against Delta, booster dose, and food for thoughts. So this will, what I can say that after this session of one hour or maybe 1.5 hours, I can promise one thing, that you all will know most of the knowledge about the corona that can be validated by the literature, by scientific community, and by everybody else around us. So definitely you are going to gain a lot actually from this session. So let us stick tuned to it, and we will dive into a couple of slides here. So on my first slide, as you can see, and we know, and most of the people know about it, that WHO declared this particular corona pneumonia related disease in 31st December 2019 and detected as a virus in Wuhan. After that, on 10th January 2020, we got the full genome sequence of that particular virus. So that's where the real scientific journey has started. Before that, it was just a mystery. We didn't know how it is happening. And 161-year-old person in that a open market in Wuhan got died. And after that, we were just like a thinking what exactly is happening. But on 10th January 2020, we came to know that there is something more to it once we have ventured into the full genome sequence of that particular virus. So after that, we looked into the disease, and most of the people know about it, that this particular disease was having a 3 phases. One is the early infection, then it get to the pulmonary phase, and then the third stage is the hyperinflammation phase. So when we looked into the particular phases, so viral phase, it settles to the pneumonia, and then the inflammatory phase. What kind of a symptoms that we used to see? Those symptoms was like a fever, dyspnea, hypoxia, ARDS, shock, and multi-system organ failure. When we looked into the clinical signs, there is a issues with the lymphopenia, hypercoagulability, lung consolidation, thrombosis, and high inflammatory markers. Those has been seen in this particular phase, where we know everybody came to know what is the C-reactive protein, what is the D-dimer, what is IL-6. So all those kind of inflammatory markers comes into that phase. So the idea was this, that, okay, if we can control this particular disease into the initial early phase where there is this stage is known as the Burmia, but later on, it becomes kind of a inflammatory and immunoinflammation. So if we can manage until like a inflammatory, generally we were able to save the patients. But if it has gone beyond that, then always there were challenges that has been faced by many clinicians. And during especially the Delta time, we have lost many people also in that process. When we looked into the symptoms, we were used to get like those symptoms started from like a day 3 and it goes on to like a day 7. And how the immunity has been responding, that initially it was like a IgA, IgM, IgG. So these all are the antibodies which is produced in our blood system, especially the IgM or IgG. IgA is like always available in our mucosal system as well. So that's how the pathogenesis of that SARS-CoV-2 was taking place, and that's how we divided into those 3 phases. Coming to the next slide, where we see that, okay, how the biology itself, the mutation, and how the symptoms were changing when there is a change into the virus itself. So old strains, what were the main features, clinical features? That the fever, persistent cough, a loss of smell or taste. So that's where everything started. Later on, there is a new strain came into the picture, and we keep adding the new symptoms as well. It can be a body ache, pains, conjunctivitis, sore throat, and the rashes on the skin, diarrhea, discoloration of fingers, and headache. So as you can see that when there are mutations, the biology is changing, and particularly to those mutations, our biology is also changing in order to tackle that particular disease. So a lot of things are happening on that front. Then what kind of testings and the diagnostic that was available to us then, and what is our scenario as of now? So as we know, in January 2020, we had only one lab in India, that was the National Institute of Virology, NIV Pune. In order to handle those kind of viruses, which was required like a biosafety level 3 or biosafety level 4 kind of a lab infrastructure. Later on in September 2020, we got like more than 2000 labs very quickly. And now these labs are like a close to like a 4,000. I don't have an exact number, but definitely more than 3,000. I read it somewhere 3,220 or so. So what we can see here that and I think we should take a pride into this in this particular moment, that the competence, capabilities, and the knowledge with the Indian scientists, the Indian community, the clinicians, the physicians, the pathologists, everybody was like a so competent enough. Only what was lacking? The only lacking was the infrastructure. The moment infrastructure came from various funding agency, including the government, we had that knowledge, and that was the only reason we were able to scale up this kind of a massive infrastructure. Because you can, if the money is provided, you can always buy the instruments, but who is going to work with those instruments? For that you need a really a great experience, competence, and capabilities. So I'm very happy to see that India is always having that kind of a competence. We were only always looking for a chance. So thanks to the government over here that made that possible, but at the same time, kudos to the Indian scientists who held on this technology, and in a very limited period of time, they have mastered the art, and they have gone beyond like a 3,000 labs, which is able to detect, diagnostic, and as well as test as many as sample they can do, not only from one technology, but by using the various other technologies as well. So coming to that particular, so what kind of a testings we are doing as of now? So as we know, because I'm not going into the detail and I try to free from many biology and science jargons over here, that but at least now it has become kind of like a common knowledge that the particular virus. So when we are doing a test for the virus, we generally look at three things that can be checked. One, we can check the nucleic acids, like the genetic material of the virus. We can check the protein that is produced by the virus, or the antibodies, those are produced by our body like a host system. So when we are talking about the genetic material, we are definitely talking about the viral RNA. So when we are talking about the viral RNA, it can be detected by the RT-LMP technology, which is a loop-based real-time PCR. And then there is a RT-qPCR, which we are using very routinely. And when it comes to the genome sequencing, then we are using the NGS. But there are some other technologies also, like a CRISPR and some of the technology to detect the other viral RNA. So what I can say that these all technologies are detecting only the viral RNA. Whereas when we talk about the antigen, so antigen is that we are detecting the protein that is on the surface or in a form of the epitome that is available on the virus. But when we are talking about the antibody, it means we are talking about the antibodies which is produced by the host like us against that particular virus. So we can do all three kind of a detection. All those techniques are available with most of the labs in India. Now the new twist has come to that particular testing, that we know that when we are testing for the COVID, we are always checking about the N gene, S gene, E gene, sometime M gene. This is what we are doing. And fourth also we do time to time, which is the RDRP gene. So when we are doing those kind of a testing, and when we detect the genes or those proteins, we say that, okay, that person is a COVID positive. What is happening in the new scenario with the Omicron introduction? So what Omicron does, as we know that in particular Omicron, there is a heavily mutated S protein. So in that particular test, when you run, it can escape this particular test. So when there is a S protein is not detected or S gene is not detected, then that becomes a suspected case of the Omicron. This is what we call as a S gene failure or S gene dropout test. And once we get those kind of a samples, then we can confirm it by the full genome sequencing of that for a confirmatory test. But definitely we can say that yes, it is most likely be Omicron. That's what we can say. Coming to the next slide, what is happening in terms of various mutations. So when we encounter the SARS-CoV-2, here as you can see that this particular virus is quite large compared to the influenza virus as well as compared to HIV. So when you look at the genetic material, that how much genetic material this particular virus is carrying, which is almost like a 30 kb. In case of influenza, it is around 13.5 kb. In case of HIV, it is around 9.8 kb. So you can see that this particular virus is almost like a 3 times bigger in the size compared to the virus that we know in that particular space, which we call as like a quasi-species. When we look that, okay, this particular virus is code for 29 proteins, whereas influenza 10 to 14 protein, HIV codes 15 protein. Now the thing is this, okay, why this is very intrigue over here, where you will understand कि ये जो particular virus है, ये virus के अंदर इतना ज़्यादा genetic material क्यों है? Why there is so much genetic material in this particular virus? Because this particular virus is having some other features also, which was never seen with the rest of the viruses in that particular space. So it is having a proofreading mechanism. So proofreading mechanism, like it is able to detect if it is making any kind of a mistake, making those long polypeptide chains, like making the proteins, if it is making any mistakes with that, it is able to proofread and almost like a clear those errors also. So it is, I call it as a like a very smart virus. ये अपने आप में एक ऐसा वायरस है जो खुद के प्रोटीन बनाने की क्षमता को पूरी तरीके से मॉनिटर कर सकता है, और नंबर ऑफ म्यूटेशंस जो इस पर्टिकुलर वायरस में होते हैं वो 0.45 म्यूटेशन पर मंथ. In case of influenza, 2 mutation per month, and in case of HIV, it is almost 4 mutation per month. So that is the simple reason that okay, it doesn't require that kind of a mutation, because when you go for more mutation, you are going to make more mistakes, or it may be like a more prone to make mistakes, and because of those mistakes, it will be a like a suicide. One mistakes can be suicidal also for the virus. But in this particular SARS-CoV-2 virus, since it is having a proofreading mechanism and can able to make the long polypeptide chain, and then having a proteolytic machinery which can cut that long polypeptide chain on the different places in order to come up with the different kind of a proteins which facilitate the various functions for that particular virus. So that's where this particular virus is very smart compared to the viruses that we are comparing with. Now I come to that, okay, how exactly this virus generally penetrate or attach to the human or host cells in this particular case. So here you will see कि यहाँ पर हमारे जितने भी cells हैं body के अंदर, जितने भी cells नहीं, there are some cells in which we are having the ACE2 receptor, and this ACE2 receptor that gives the opening to that particular virus. So this virus generally use that ACE2 for the entry. So it comes into the contact of ACE2, it fuses together and deliver that particular viral RNA inside, and that's how later on it makes a lot of copies, and then there are a lot of new virons are made. So that's how it is able to penetrate in our host cell system. What exactly is happening, and those kind of a ACE2E expression is like a mainly in our lungs, in our heart, in our gastrointestinal tract, as well as into the liver. So what generally happens that wherever there is a ACE2 receptor is expressed in our body cell system, this virus can easily go over there, starting from the lungs and so on. And that is the reason that many patients which did not able to fight well, maybe in a lack of treatment or their biology did not support them so well enough. So what exactly happened that, okay, those kind of a cases like a lead to the multiple organ failure also. The reason was that this virus was good enough actually to penetrate in all the organs or the various cell types which was expressing the ACE2 receptor. So now I come to the comparison with other RNA virus, how unique it is. So as we know that, okay, now we are following that particular Greek alphabet system. Is system ke according now we are giving them the names also. So we know that there are total 5 variants of concern that has been detected so far. So why I am bringing that up? Because you will see that mainly into the media as well as from some other places, you will find that every day they are coining a new word and they are coming up with every time with a new buzzword in the name of a new virus, new strains, new variant of concern. So there it will definitely like create a panic into the system. So what I mean to say that until or unless the WHO has not classified them the variant of concern or variant of the high consequences, until that there is a then there is a only for the research purpose, but it may not be useful for the public per se. So in that particular way you can say that we know about the alpha variant, we know beta, gamma, and we have the here the timeline also that on which particular timeline actually those particular variants appeared. So gamma, delta we know because we faced it very well, then so on. And after that, we have the Omicron. So 1, 2, 3, 4, 5. These are the 5 variant of concern. Rest of the things will always like a get analyzed because we are going to do the genome sequencing into the public. We will like a try to map actually those sequence with the existing virus profile that we have, gene profile we have. If there is any change, we will report it, or the scientists will report it. They will deposit that particular sequence to the WHO and some other database. But it doesn't mean that, okay, that there is a something for the public and they need to worry about it. If there is a something need to be done, we will hear that. So that's how that we have adapted actually that Greek alphabets actually to name those particular virus for various regions that we did not like a name this new. WHO said that, okay, new like it creates the confusion with the new NEW, and G, we know the obvious reasons that has been talked a lot more into the newspapers that why they don't want it to have the G. So we directly went to the Omicron here, and then it will follow the rest of the variants. So we have the variant of interest, variant of concern, and variant of the high consequences. Coming to the Omicron. Now, what is happening with the Omicron? This is very interesting. So in the Omicron, as we know that there are almost like a more than 50 total mutations, जिसमें के करीब 26 के करीब 26 जो म्यूटेशंस हैं वो आपके स्पाइक प्रोटीन के ऊपर है. यहाँ पर ये एक जिसको हम वेन डायग्राम बोलते हैं, उसके अकॉर्डिंग यू कैन अंडरस्टैंड कि हमारे साथ में डेल्टा के साथ में इसमें कितने हैं, गामा के साथ में कितने हैं, बीटा के साथ में कितने हैं, अल्फा के साथ में कितने हैं. तो जो आपको बीच में सर्कल्स में आ रहे हैं, those are the common ones, but you will identify there are a lot more which is sticking out from that Venn diagram. Those were like a really unique. So as you can understand कि जो ओमिक्रॉन जो पर्टिकुलर वायरस है, जो वेरिएंट है, ये अपने आप में बहुत यूनिक है कंपेयर टू डेल्टा, कंपेयर टू बीटा, कंपेयर टू अल्फा और गामा. अभी इसके पीछे एक बहुत अच्छा साइंस है. वो साइंस क्या है कि अगर आप समझेंगे कि यहाँ पर कहीं पर आपका जो है अल्फा शुरू हुआ, इस लीनिएज के अंदर बाद में जाके डेल्टा मिला, इसी लीनिएज में जाकर के बीटा मिला, गामा मिला, और ये सारे म्यूटेशंस बीच बीच में होते रहे। ये क्या है, बट ओमिक्रॉन सडनली appeared actually. Nobody knew about it, that okay, what is happening with this, because this is a completely new lineage. It is not like a common with the other variants that we have seen earlier. तो ये जो ओमिक्रॉन है ये एकदम से अपीयर हुआ, तो इसके पीछे अभी बहुत सारा रिसर्च हुआ है जिसमें ये जानने का कोशिश किया गया है कि इतने टाइम के अंदर ये ओमिक्रॉन था कहां पर। क्योंकि अगर ये एग्जिस्टिंग अगर सिस्टम के अंदर अगर म्यूटेशंस हुए होते थे तो वी शुड बी एबल टू गेट फ्यू मोर म्यूटेशंस ओवर हियर, but there was no mutations at all in this lineage, and after 18 months suddenly a new variant has appeared. What has happened to it? And that is the reason that it is not so common. It is very different compared to all the viruses that we have seen in that particular space. So I will come back to that little later, and we will try to decipher that what the science says about it. Now, looking at the Omicron side from the case study in South Africa. So, because as you can see that, okay, the trend is going to be followed like a South Africa, then UK, after UK it is US, and after US it is the East, like Eastern countries, Europe and India and so on. So we believe that if we are able to understand the trend from west to east, we can prepare ourselves. So when we look at the South Africa in that particular age group, what we were getting in Delta and what we are getting into the Omicron. So one good news is this, that in this particular Delta Omicron, that there is a pathogenicity is something which we can talk about it, because this particular Omicron virus is not so pathogenic. Coming to the even into the death that those has been reported in those various waves. So wave 1, wave 2, wave 3 and wave 4, and wave 4 is they are talking about the Omicron. And this red line you see over here, this is giving you the number of deaths. So it is almost like a no death kind of a scene in South Africa. There have been maybe some deaths because of other comorbidity regions, or maybe if the patients is having a immunocompromised patients living with the AIDS, so there may be some issues with that. The people who were like a vaccinated or having a hybrid immunity, vaccinated and got natural infection, those kind of a people, they have not seen too many issues with their health, and there was not much like a hospitalizations or followed by the ventilator or intubation or any kind of a death that has been seen in particular in South Africa. So that's a good news over there. Coming to the clinical symptoms of Omicron. So I have like a captured it from the various data, where actually I have looked more into the UK data because their data looks quite good. So 65% people almost like reporting that they are having headache. 57% people are reporting that they are having a scratchy or sore throat, or 55% people reported sneezing. 65% people body ache and fatigue, weakness, and runny nose or stuffy nose, 65%. There were very few cases which were talking about the fever, which is like a high grade. When I say high grade, like a less than 100 degree Celsius, not those many, not that many cases. So not 100 degrees Celsius, 100 Fahrenheit. Fever and loss of smell, these were not so common actually in those Omicron patients. So now we can talk a little bit about the age Omicron interestingly less pathogenic. We can expect very large number of cases. Symptoms may be mild, that we know. From the vaccine front, we know that still we have the some of the immunity because our T cells seems to be responding. When we say T cells are responding, it means that they are able to recognize the epitome that is present on the antigen from the virus. Or we also know that Omicron infects and multiplies 70 times faster than the Delta in human bronchus. When we say bronchus, we are totally talking about the upper respiratory tract, and 10 times slower in the human lung tissue. So this is a good news over here. And there will be the lesser lung inflammation because there is the multiplication into the lungs is very low. So if there is no lung inflammation, there may not be the pneumonia, there may not be the ARDS, there may be not any kind of a any kind of a like a arrhythmia or those kind of issues, or any kind of a lung consolidation, which we used to see in the case of Delta, where everybody was running for the high resolution CT scan and so on. So this is what the pathology, the pathogenesis of the Omicron is totally changed in that way. So now, what exactly was happening in case of Omicron or in case of like a Delta? तो इसमें इस कार्टून के माध्यम से आप ये समझने का हम कोशिश कर सकते हैं, जिसके के बारे में हमने बात किया कि जनरली जब पहले ये हमारा एक ह्यूमन सेल है, इस सेल के ऊपर जब भी ये वायरस आता था तो इट ऑलवेज लाइक ए यूजिंग दिस ACE2 साइट एंड बाइंड टू दैट पर्टिकुलर थिंग. सो this binding was happening through the another protein, membrane protein that we call as a tempress. तो डेल्टा के केस में क्या होता था कि ये जो टेम्परेस प्रोटीन है, इसको हम प्रोटियोलिटिक प्रोटीन भी बोलते हैं. प्रोटियोलिटिक का मतलब है इट हैज ए एबिलिटी टू कट द प्रोटीन्स. तो ये क्या करता था कि जिससे कि ये वायरस बड़े आसानी से इसके साथ में फिट हो जाए, तो ये यहाँ पर इसको फिट करने का काम करता था. इट चॉप आउट द पर्टिकुलर प्लेस in this particular virus so it can fit properly. तो जब ये properly fit हो जाता था तो it has given the entry to the system and it was very efficient in term of the pathogenesis. तो बड़ी आसानी से जो है इन्फेक्शन को पैदा कर सकता था. इस प्रकार से जब ये फ्यूज हो गया तो फ्यूज होने के बाद में it can deliver that viral RNA into our cells and that's how it can make them sick. और इसके बाद में ये बहुत सारे दूसरे छोटे छोटे मतलब छोटे छोटे से मतलब है दूसरे वायरस को बना सकते थे. तो अब तक डेल्टा के केस में क्या हो रहा था कि देयर वाज ए लाइक ए एफिशिएंट एंट्री वेयर वन ऑफ आवर प्रोटीन वाज हेल्पिंग टू मेक दैट एंट्री, और उस एंट्री की वजह से इट वाज एबल टू हैव ए सीवियर पैथोजेनिसिटी and it can give us the severe disease also. So this is what was happening in case of the Delta. Now what is happening in case of Omicron? Why the pathogenicity is less? So one reason is this, that now this Omicron has been mutated in such a way so that it doesn't require this tempress. So now the tempress is just asking, okay, guy, I was like used to be really useful to you. What is happening? Why don't you use me? I will make you fit over there. And Omicron says, okay, I don't need you. I am better off than you. I can deal with it. And that's where the Omicron is able to infect and master the art actually to enter into the cell. But at the same time, it has favored us in a way. How it has favored us? That कि जब ये वायरल RNA जब हमारी सेल्स को जब इन्फेक्ट करता था, तो there are many cells which goes in our lungs for the syncytium formation. सिंसिटिया फॉर्मेशन में क्या होता है कि हमारे जो एल्वोलियर सेल्स जिनसे कि हमारा लंग्स बना हुआ है, वो सारे के सारे जो सेल्स हैं वो आपस में जुड़ करके फ्यूसोजेनिक एक्टिविटी के अंदर मिलकर के एक जाइगेंटिक सेल बना लेते हैं, जिसको कि हम सिंसिटियम फॉर्मेशन बोलते हैं. उस सिंसिटियम फॉर्मेशन की वजह से क्या होता है कि हमारे लंग्स के अंदर जो ऑक्सीजन को बाइंड करने की क्षमता है वो कम हो जाती है. बाद में अगर डिजीज प्रोग्रेस होती है तो फाइब्रोसिस में कन्वर्ट हो जाता है, जिसकी की वजह से ऑक्सीजन डिप्राइव हो जाते थे पेशेंट्स, एंड वी नीड टू आइदर नीड टू पुट देम ऑन द ऑक्सीजन और वी नीड टू पुट देम इनटू BiPAP or any kind of a need to be do intubation, ventilator, and all those mechanics comes into the picture when you see that the disease is settling down into the lungs. तो यहाँ पर क्या हो रहा है कि ये जो टेम्परेस जो है, ये जो पर्टिकुलर प्रोटीन है, ये होता है सिलिएटेड सेल्स के अंदर। तो हमारे लंग्स के अंदर जो सिलिएटेड सेल्स ना होने की वजह से, कम होने की वजह से, सिलिएटेड जो सेल्स होते हैं यानी कि एक तरीके का माइक्रोस्कोपिक हेयर होता है जो कि हमारे ब्रोंकियल सिस्टम के अंदर तो ज्यादा मिलता है, बाकी जगह में कम रहता है। तो उसकी वजह से ये जो वायरस जो है वो हमारे लंग्स में जाकर के सिंसिटियम फॉर्मेशन को एक्टिवेट नहीं कर पाता है, जिसकी की वजह से हमारी जो लंग की जो पैथोलॉजी है वो डिस्टर्ब नहीं होती और हमारे लंग काफी हद तक जो है सुरक्षित रहते हैं। तो ये एक जो चेंज है जो हमारे फेवर में हुआ है ओमिक्रॉन के म्यूटेशन की वजह से है क्या है। तो अभी नाउ एज यू कैन सी इन द अनदर कार्टून यू विल फाइंड दैट पहले क्या होता था, टू मेक इट सिट प्रॉपरली ऑन दिस पर्टिकुलर सेल, दिस टेम्परेस वाज हेल्पिंग इन कटिंग, चॉपिंग एंड सेटलिंग इट डाउन। लेकिन अभी क्या है कि दिस इज नॉट यूज्ड बाय द ओमिक्रॉन एंड इट हैज मेकिंग ए एंट्री इनटू द सेल, but at the same time it has mastered the art into entry, but at the same time pathogenicity went down because the mode of action, the mode of pathology has completely changed in case of Omicron. Now it is further like a validated, as you can see over here, that when we are talking about the bronchus, we are talking about the upper respiratory tract. And in this particular upper airway, so the entire replication of the virus is happening on those kind of sites, but not into the lungs. And this is what the problem with the Delta, because Delta was replicating into the lungs, whereas this Omicron is replicating mainly into the bronchus. And when we are talking about the bronchus, so bronchus is generally present in our body, जहां पर के हमारा जो बाहर का जो एनवायरमेंट है वहां से जो भी चीजें आती हैं उन जगह के ऊपर जनरली ब्रोंकस है। जैसे ब्रोंकस आपके लीवर में नहीं है, लंग्स के अंदर है, इंटेस्टाइन के अंदर है, हमारे नोज के अंदर है, माउथ के अंदर है। तो जहां पर एनवायरमेंट का डायरेक्ट प्रभाव होता है या एनवायरमेंट की चीजें डायरेक्ट हमारी बॉडी के साथ में जुड़ती हैं, वहां पर जो है जनरली ब्रोंकस या म्यूकोसा, जनरली म्यूकोसा रहता है। ब्रोंकस जस्ट ए पार्ट एक्चुअली टू डिफाइन दी एयरवे ओवर हियर। तो उसकी वजह से जो हमारा जो म्यूकोसा है उसके अंदर IgA एंटीबॉडी जो कि हमारी बॉडी में 3 से 5 ग्राम हमेशा बनता है, रोज बनता है, इट गेट एक्टिवेटेड ऑन द day 1. So that also helps us actually to complete the cycle of the immunology, because IgA is getting active on the day 1. IgM and IgG bit comes later because it comes from the humoral innate immunity and it is comes from the blood and the plasma. But whereas the IgA is active on day 1, so that's how we can control the pathogenicity and the severity of the disease that is inflicted by the Omicron. And when we looked more into the data, this is the data from the South Africa from Professor Alex Siegel lab, where he has clearly seen into the bronchus that it is multiplying 70 times faster, Omicron, compared to Delta, but it is a 10 times slower in lungs. So that also validated the theory that we have around that it is not really infecting our lungs. Then there is another paper that clearly suggests from that particular lab that the somehow that whoever is getting the Omicron infection, तो उस केस में अगर ओमिक्रॉन के अगर हम इन्फेक्शन को देखते हैं तो ओमिक्रॉन से जो भी इन्फेक्ट हो रहा है उसके अगेंस्ट जो एंटीबॉडीज बन रही हैं तो उनकी ये कम से कम वो 14.4 ज्यादा एंटीबॉडी बन रही हैं ओमिक्रॉन के प्रति। तो यानी कि हम ऐसा मान सकते हैं एक लेब एंड लैंग्वेज के अंदर कि जिसको ओमिक्रॉन का इन्फेक्शन हुआ है उसको शायद दोबारा से ओमिक्रॉन का इन्फेक्शन ना हो, और अगर किसी को डेल्टा का इन्फेक्शन हो रहा है तो उस केस में भी इट इज प्रोड्यूसिंग 4.4 टाइम्स इम्यूनिटी लाइक एंटीबॉडीज मोर अगेंस्ट द डेल्टा। तो कहने का मतलब क्या है कि जिसको ओमिक्रॉन का इन्फेक्शन है उसको डेल्टा नहीं होना चाहिए, और शायद उसको ओमिक्रॉन का इन्फेक्शन भी दोबारा नहीं होना चाहिए, if this data is true. So, and I believe that the way the researcher has done that work, they have done a really good work to actually prove this point. Coming to the next slide, when there is a lot of discussion around के जो पर्टिकुलर जो हमको कोविड का वैक्सीन बूस्टर लेना है या नहीं लेना है या क्यों लेना है, तो जैसा कि हम इस पर्टिकुलर पैनल में देख सकते हैं कि जब हम फर्स्ट शॉट लेते हैं तो हमारा जो प्रोटेक्टिव जो इम्यून थ्रेशोल्ड है वो आपको ये जो डॉटेड लाइन दिख रहा है ये है, तो इसके ऊपर चला जा रहा है, तो इसका मतलब है वी आर गेटिंग द प्रोटेक्शन। सेकंड शॉट लिया तो इट बिकम्स लाइक ए स्ट्रॉन्गर रेस्पॉन्स, but within there are many study which clearly says that this antibody is waning out with the time. So within like a 3 to 4 months, some people say 3 to 6 months based on their data, these antibody levels are coming down. So your second dose is somewhat it is coming to below this particular threshold. तो जब ये इस थ्रेशोल्ड के नीचे आ गया तो इसका मतलब है कि it won't be able to protect you anymore. But now we are talking about the booster dose. तो जैसे ही आप बूस्टर डोज देते हैं तो बूस्टर डोज जनरली अगर इम्यून रेस्पॉन्स देखा जाए तो एक 10 to 14 डेज के अंदर बूस्टर डोज इज एबल टू टेक योर एंटीबॉडीज टू द पीक व्हिच इज ए इवन स्ट्रॉन्गर रेस्पॉन्स, एंड व्हेन इट इज ए सच काइंड ऑफ ए स्ट्रॉन्ग रेस्पॉन्स then definitely it is going to protect us again for some time. So what it suggests that it may also come down after some time, but at least it can protect us as of now. अभी कल का ही रिपोर्ट है, ग्लासगो यूनिवर्सिटी यूके से है, क्या है, दे हैव क्लियरली मेंशन्ड इन देयर पर्टिकुलर पेपर कि दो डोज से आपको कोई खास इम्यूनिटी मिलने वाला नहीं है, ठीक है ना। तीसरा डोज होने पर आपको पार्शियल इम्यूनिटी मिलने वाला है। जितने भी वैक्सीन हैं उनका लगभग जो इम्यूनिटी जो प्रोटेक्शन है इट हैज केम इनटू द रेंज ऑफ लाइक ए 30 to 40%, बट विद द बूस्टर डोज it can go somewhat 70 to 80% depending upon the various literature that is available as of now. So do we need booster? Yes, definitely we need booster because that's what the data suggests. And we also need to keep in mind that when we are talking about the vaccines are not actually meant actually to stop the infection. It is more about like a not having too many hospitalizations, not getting severe diseases, or stopping complete the mortality. So this is the job that vaccines are doing, and it is doing wonderfully. So I think that we should not like hesitate in taking the vaccine if it is not taken, or if there is a booster dose is available, we should go for the booster dose as well. So coming to the another point, so there is a all looks like there is a everything good about this Omicron as of now, but there are some of the researchers, they are also talking about that okay, it could be the evolutionary mistake that is made by the virus itself. How evolutionary mistake? That there is a like a study, in that study they try to figure it out that why suddenly this Omicron appeared the way like I have just recently described on this particular slide. So from here to here there is nothing. How suddenly it came? So there is a study that has been done which clearly suggests that it means that this Omicron virus was not in the human. तो स्टडी सजेस्ट कि ये जो ओमिक्रॉन का जो वायरस है अगर ये ह्यूमन में है तो इसके केवल 3 रीजन हो सकते थे। नंबर 1 बात कि ये क्रिप्टिकली स्प्रेड कर रहा था और हमको पता नहीं चला, ये तो एक हाइपोथेसिस हो सकता है कि ये किसी तरीके से किसी पॉपुलेशन के अंदर म्यूटेट हो रहा था और हमको पता नहीं चला इसका। दूसरा ये हो सकता है कि कहीं पर कोई इम्यूनो कॉम्प्रोमाइज्ड अगर कोई ऐसा पेशेंट्स हो, उन पेशेंट्स के अंदर जो है क्या है ये धीरे धीरे करके म्यूटेट कर रहा हो एंड यूजिंग देम एज ए होस्ट। but there is a too much difference within their mutations. So it is very difficult to say if it would have been in the human. So the data and the research says कि ये जो ओमिक्रॉन है ये लगभग अप्रैल और मई 2020 के आसपास ही somehow there is a interspecies evolutionary jump. Interspecies evolutionary jump means that कि ये ह्यूमन से वापस किसी एनिमल में चला गया, और वो एनिमल जो बताया जिसके बारे में, that is gone into the mice. When we do that control, that entire study, तो हमको पता चलता है कि जो ओमिक्रॉन के अंदर जो स्पाइक प्रोटीन में जितने भी म्यूटेशंस हुए हैं, those mutations are perfectly matching, not matching, like it is making it able to fit particularly with the ACE2 receptors of the mice. तो एक तरीके से ये ह्यूमंस से दोबारा से माइस में चला गया, माइस में जाकर के इतने सारे म्यूटेशंस हुए, क्योंकि जैसा अभी हमने बीच में देखा कि जनरली एक मंथ के अंदर 0.54 म्यूटेशन के करीब हो रहे हैं, और इस वायरस के अंदर 50 म्यूटेशंस के ज्यादा हुए हैं। तो ये 50 म्यूटेशंस एक बार में नहीं हो सकते, तो कम से कम इसको 18 महीने का समय लगा है इन म्यूटेशंस को एक्वायर करने में। और ये एक तरीके का इवोल्यूशनरी मिस्टेक ही हो सकता है कि जहां पर के एक वायरस ह्यूमन से वापस चला गया माइस के अंदर, और वापस से इंटरस्पीसीज जंप हुआ कि ये माइस से निकल करके वापस ह्यूमन में आ गया। सो इन ए वे इट इज इन आवर फेवर, but it can happen anytime or vice versa. That's where we should not let our guard down, and we need to make sure through the genomic surveillance in the country, sero surveillance, making sure that if there is any new virus or new variant is appearing, we can catch it first rather than before it will spread into the entire community and make all of us sick. So that's why the genomic sequence is very important tool that we should use quite often. And coming to my almost like a last slide, so where I just request all of you that these are, if nothing will work, okay, as I always say, mask is always going to work. Wear your mask, sanitize your hands, and avoid the crowd space. And I wish you all thank you and stay safe. So that's in a summary, and now I am open to any kind of a question that you guys may have. Thank you so much.
That's wonderful, Dr. Upadhyay. In the chat box, I can see there are two questions by Mr. Anil Saxena. The first questions before you. Dr. Upadhyay, sir, as per media, many were affected by both Delta and Omicron virus. In this case, which virus will dominate more?
See, there is it is quite evident from the US data, Australia and Scotland, and as well as into South Africa also, that Omicron is replacing the Delta. So ultimately, you will find that almost like a 90% cases that is coming in US, those are becoming the Omicron cases. So I can comfortably, I can say that Delta is going to be replaced by Omicron, and Omicron is going to be the dominant in that particular space. But this is a natural phenomena where the co-infection can happen. When there is already a one dominant virus in the host, one new virus or variant comes into the picture, they can just going to infect in a one particular cell, and then their genetic material can get mixed, and based on that co-infection, you may get a new variant. But so far there are many variant has been reported, not like a Delta Cron only. There is a Florena, there is a IHU from France, and there is a something from Israel also, now another one from the Cyprus. So this kind of a things, it is keep coming. But right now it is, and which is a natural phenomena we have seen earlier also. So for a general public, they don't need to worry about that part.
डॉक्टर साहब, मिस्टर अनिल सक्सेना का ही दूसरा प्रश्न है कि कुछ वैज्ञानिकों का कहना है कि ओमिक्रॉन जो है वो इसका समापन करेगा, इसका खात्मा करेगा. व्हाट यू हैव टू से ऑन दैट?
हाँ, ये एक बहुत अच्छा एक्चुअली थ्योरी इवॉल्व हो रहा है, क्योंकि मैं तो खुद मैंने भी एक दो बार बोला है एक दो जगह के अंदर के ओमिक्रॉन इज बिकमिंग ए काइंड ऑफ ए नेचुरल वैक्सीन. तो अभी हो क्या रहा है कि ओमिक्रॉन जनरली हम वैक्सीन के द्वारा भी क्या अचीव करना चाह रहे हैं, वैक्सीन से भी हम ये अचीव कर रहे हैं, वी आर गिविंग देम ए वेरी कंट्रोल्ड इन्फेक्शन एंड ट्रेनिंग द बॉडी ओके विद नो ऑलमोस्ट नो पैथोजेनिसिटी. ओमिक्रॉन के क्या फीचर्स है? ओमिक्रॉन के फीचर यही है कि दिस इज ए लाइक ए मिनिमम पैथोजेनिसिटी, but at the same time it is able to train our immune system. So later on what can happen if the Omicron is going to replace all the data, all the Delta variant, then the only dominant virus will remain will be the Omicron. And Omicron is not so pathogenic. And at the same time, the data suggest that Omicron cannot infect you again if you have already got infected by the Omicron, because it is producing 14.4-fold more antibodies against it. Delta cannot come back because Omicron is giving you 4.4-fold antibodies against the Delta. So that's where we can say, okay, I can say it based on the data. But as I always say, we need to take everything with a pinch of salt, and we are hoping that, okay, it will found true, but we will come to know maybe in a few months from now that which way it is going. But what Dr. Saxena is saying, that is absolutely true. There is a lot of research that can prove that point.
Thank you, Dr. There is one more question by Swati Rajiv, I can see in the chat box. Here it is. Sir, if we are having any of the symptoms as a sneezing or coughing or body ache, when do we need to go for the tests, and which day will be counted as the 0 day?
Yeah. So generally जो फीचर्स का हमने बात किया है, तो आप देखोगे वो सारे फीचर्स में करीब करीब जो 65% लोगों के जो फीचर्स हैं, ठीक है ना, वो ज्यादातर कॉमन फीचर्स हैं. तो जिसमें ये फीचर्स हमको मिल रहे हैं, जिसमें कि एक हेडेक जो है वो बहुत ही प्रोमिनेंट फीचर है. तो अगर किसी को हेडेक हो रहा है और उसके साथ में कुछ ऐसे फीचर्स भी आ रहे हैं, तो डेफिनेटली इट विल बी ए केस ऑफ कोविड. आई एम नॉट सेइंग वेदर इट इज ए ओमिक्रॉन और नॉट, बट डेफिनेटली दैट पर्सन मे हैव ए कोविड. सो द मोमेंट दैट पर्सन इज गेटिंग दोज काइंड ऑफ ए फीचर, maybe day 3 will be the appropriate actually to go for the RT-PCR test or any other test in order to confirm it, because the incubation period for the Omicron is also considered to be 3 days. Keeping that into the mind, the WHO and the CDC has reduced the quarantine time also from 10 to 14 days to now 7 days. In some cases it is 5 days also. So I think that day 3 will be, I think, something that we can consider actually to go for the testing.
Doctor, I have a question.
Yes.
This is about booster. You mentioned that booster may be required after 3 to 4 months, but the government here has recommended 9 months. That is one.
I also had to raise the same question.
And the other one is that will a second booster or a third booster would also be required, because you showed a graph that it keeps on increasing.
Yeah. So what I can tell you that in Israel, you see that they are already with the 4th booster actually, like a 4th dose.
So
Okay.
you can have any number of boosters.
Yeah, so that's what it is going on. Like it has become, this is what I was saying from the very beginning. As you know, that these are the what you call them, these all are the antibody based vaccines. It is not a B cell based memory cell based vaccines, so their memory is going to be very short. So at the moment the concentration or the titer of those antibodies come down, we are again like a prone to catch that infection. So how much is that appropriate to keep pumping those spike protein into the body, keep getting those antibodies, how long it is sustainable, how long we need to do it? It is very difficult to say, but right now we are fear for today, not for tomorrow. With that approach, I certainly see that there is an advantage of getting third dose or fourth dose. There is a COVID Chronicle that has published just yesterday. They have clearly said, okay, 2 dose has no effect. Okay, researcher can say that, media may not say it, but researcher has clearly said that, okay, there is a no advantage. 2 doses is no dose. Okay, the third dose is providing you the partial protection. Fourth dose, maybe it will give further. So it's a continuous story that is going on. But what I am stating based on the facts, actually, based on a lot of literature.
What about a medicine? Is it not coming? A medicine is supposed to stop process, isn't it?
Yeah, so medicine is something like, again, when there is just so much mutations, as you know, that the particular medicines are also designed based on a particular active site into the particular receptor when we talk about the drug target mechanism. So when something is changing so dynamically, it is very difficult if it is not conserved region in that particular virus to target it. Although I think that Mpro protein inhibitor that is coming from Pfizer, and then there is a one drug, a small molecule is coming from Merck. They may have some effect, but how much effect it will have, it is very difficult to say. And then when we are going to introduce those small molecules, then we also need to keep in mind that there will be a lot of drug-drug interaction issue also, because those drugs are going to be very different. So let us hope for the best if they will do the job, but I am not so optimistic about that.
Well said, Doctor. Well said. One question by Mr. S.K. Verma, as he says, shall we start the Ayurvedic medicines as we did in the first phase?
See that, okay, I am a kind of a believer of Ayurvedic medicine. Let me put that disclaimer here, because especially when it comes to the communicable and infectious diseases, okay, this is all the game of your immunity. And most of the Ayurveda principles and the drugs are generally immunomodulators. Okay, so if we can find something which is really working, we can definitely use that. I am not against it, but I think the research also need to be done in such a way where we know that this is something works. In name of Ayurveda, we should not start pumping us with anything else.
Yeah, thank you.
करेक्ट डॉक्टर साहब, अभी जैसे जो पॉजिटिव आ रहे हैं केसेस, तो उसमें ये आइडेंटिफाई नहीं करके दिया जा रहा है रिपोर्ट्स में कि आदमी को या पेशेंट को ओमिक्रॉन है या डेल्टा है. हाउ टू आइडेंटिफाई दैट पार्ट?
या see, this is something can be done, but maybe for a patient it is not so important. It is more important for the government and the health agency to understand and devise their strategy towards the management of that particular disease. But whereas I can say one thing over here, that as I mentioned that there is a N gene, E gene, S gene, and the RDRP gene. Okay, this is 4 genes that we are testing in our COVID protocol. So when there is a S gene dropout, okay, when your system is, when your RT-PCR is unable to detect the S gene, then this may be most likely a Omicron case. But the government has adopted a policy, I think, in India, that they are only taking 5% of samples for the full genome sequencing in order to confirm whether it is a Omicron or something else. So this sample of 5%, to me, it is really a low number in order to get the clear picture. And that picture may also not help us to make some strategy, especially the health related strategy in country as of now. What I know that I live in Sweden, we are doing almost like a 20% UK is doing 20%, nobody is doing 100% for sure, and US is doing somewhere 25 to 30%. So to get a kind of a sense that we are still getting more prominent or dominant is the Omicron. But if something will change within the community, maybe some new variant will appear, it will be very difficult to detect it by us. I don't want to say that, okay, it may become like another Delta kind of a situation where the UK was able to figure it out that there is a Delta in Indian population, whereas we took a lot of time actually to understand that there is a Delta variant is in process in India.
Dr. What about 9 months or 4 months? Yeah.
That was a question.
Yeah. So what I can say that, okay, most of the literature that is saying 4 to 6 months. Okay. Let us set that range. I can say that, okay, I should not say, but I am a well-read person actually on that aspect. And I have not seen anywhere, any science or any university, any research group is reporting that you should take it after 9 months.
9 months.
Oh, the government is saying after 9 months.
Government of India has recommended for 9 months, for gap
Yes.
of 9 months.
Yeah.
They
So
will
generally,
not
I
give
would
a-
say
They will not
that
give a booster before 9 months.
yeah, I think they should go for a 6
Review.
months. That's all we can request. But ultimately, they are going to decide. They are in that position to make the decisions.
But we feel it may be a matter of availability also, Dr.
Yeah, could be, but they need to give that region. But they should not like a fool around the science actually.
Correct.
Today's Dr. Saab session, let him have the final say.
Dr. Saab, I have one practical question.
Hello?
Pankaj here.
Hello?
Yeah.
Hello?
Hello.
Dr. Saab, there is one more question regarding the booster.
Yes.
Should it be the same vaccination or a different vaccination?
Yeah, that's very interesting actually. So in case of India, in fact, we have not done that kind of a study. Okay, whether it is a Covaxin or whether it is a Covishield. Okay, but definitely they have in US, they have included that Novavax that is coming as a Covax in India, and Novavax it is known in US. So what they have done that they have studied the 3 J&J vaccine, Moderna vaccine, Pfizer vaccine,
Yes.
and they have like a in the various permutation combinations, they have given the boosters. So if J&J is taken, then they are giving Moderna vaccine. If Moderna is taken, they are giving Pfizer, or if Pfizer has been taken, they are giving J&J. So based on that, they have made those categories where they found that mixing of these particular vaccines is really good because they see that there is an advantage because the neutralizing ability is increasing, and at the same time the production of the antibody also increase. So that's where they see that there is a benefit, there is an advantage of mixing up those vaccines. But right now the situation that we have, like we say that beggars can't be choosers. So I would say that whatever we are getting, okay, let us take
We
it
should get
and
it.
keep going.
Correct.
Good.
So over to Pankaj now. Yes, Pankaj.
Dr. Saab, in Lesotho, you have told that they have not used antibiotic in treatment of this COVID, while in India, everyone is using azithromycin. As there is sore throat or suspected viral fever, everyone is recommending, and if no one is recommending, the people think that they are not getting the proper medicine. In such cases, what to do? Because antibiotic is for, as for we know, bacteria, and this is the case of virus.
Yeah, see, first of all, let me put a kind of a disclaimer here that I am not recommending any medicine. Okay, that is number one. The thing is this, okay, generally the clinicians always have that discretionary power and they can make the decision what they feel it is best for their patient. So if the clinician feels that somehow the azithromycin is going to help their patient, they can always make those kind of a prescription. And especially when we talk about the the scratchy throat or sore throat, okay, always we go for the erythromycin, azithromycin. And living in Indian condition, there is a most of the time there is a throat infection. So they may be like a suspecting that, okay, maybe there may be some like a viral symptoms, but at the same time, there may be some like a bacterial infection of throat. And that could be the reason that they may be prescribing. So I'm nobody to say yes or no to it, but I think that the clinicians are the best who can decide in favor of the patient.
That's wonderful, Dr. Sir. Just one question, layman question, rather. In India, this is being talked a lot. People are saying and quoting that the booster is also the same thing which was given as the first dose or the second dose. Is there any difference in the dose, any kind of different thing, or the same is being repeated? What's your say?
Yeah, it is same being repeated. Only
Okay.
there is a little difference in Pfizer and Moderna, which is not actually available in India. Their dose is different. Like a Moderna is dosing at like a 50 mcg, and whereas the Pfizer is dosing 100 mcg, where earlier the both were having like a 100 mcg, but Moderna found that 100 giving 100 mcg is having some more like a side effects, or so that is the reason that they have reduced their dose to 100 to 50 mcg. But rest
Okay.
of the vaccines are just same. There is a no change. Only the Pfizer is coming in March, which is specific to the Omicron. So Pfizer is coming with a new vaccine in March that will be specific to the Omicron. But otherwise, rest of the vaccines are same, which we have taken earlier.
डॉक्टर साहब, एक बार आपने कहा था कि ओमिक्रॉन इस महामारी का अंत करेगा, और ओमिक्रॉन को लेके गवर्नमेंट लॉकडाउन लगा रही है, बहुत सारे फियर है मार्केट में। तो अगर ये अंत करेगा तो इसको लेकर इतना डरावना माहौल क्यों?
नहीं, डर के रहने का जरूरत नहीं है, बट प्रिपेयर रहने का जरूरत है, अलर्ट रहने का जरूरत है,
प्रिकॉशन लेने की आवश्यकता है।
हाँ, हमारा pragmatic approach होना चाहिए. I also believe that this Omicron could be the way out from the pandemic. Okay, it may get to the endemic stage. I also believe that, and it is not because we are clever. Okay, it is not because we are clever. It is only because, as I said in my opening, that the Omicron has a evolutionary mistake that has become kind of a boon for us, that Omicron has gone into the mice, mutated over there, came back in human again, and that's how it happened. So if we are looking at natural phenomena, okay, if being a human being, if we are not yet humble, then I really do not know that what we are talking about. So I also believe in that, but we cannot let our guard down. Let us hope for the best, that all the statements that if I have put it out or some other people have put it out in the best of their interest and intent, they will found true going forward. So we can all hope for that.
But nature का auto correction मानिए?
Absolutely, you can say that, yeah, as of now.
I feel it, Dr. Saab, I feel it has been a wonderful session. I can see many compliments from you for you in the chat box. You too might have observed those. And for finally, if there is any more question from the audience, I will be here. Before I request Mr. Khan, the core group member of Lucknow Management Association, to kindly propose a formal vote of thanks on behalf of Lucknow Management Association and the YMF.
Thank you, Dr. Upadhyay.
ने ये कहा था कि ओमिक्रॉन वैक्सीन है खुद ही, तो फाइजर की वैक्सीन का है कि नहीं, वो तो न्यूट्रलाइज कर देगा इसका इफेक्ट.
Yes, that is true. I was also thinking, what is the business strategy behind it? Why are they making that Omicron specific vaccine? So I also did not understand that why they are doing it, but that's the scenario here.
Business gimmick, isn't it?
Gaurav ko bulao.
Sorry?
More money.
Gaurav Devesh. That? खुलवा देना, वही पूछ रहा हूँ। Yeah, Khan sir, please carry on.
मैंने कहा शायद एक आध और कोई और इंटरजेक्ट कर जाए बीच में से पिछली बार की तरह. So let's wait for a second. Is there any
Sure.
other question?
Sure, please.
Seems now
Any other
there is no
question, please?
एक ही क्वेश्चन है डॉक्टर साहब, अभी ओमिक्रॉन अभी डेल्टा क्रॉन के लेकर जो न्यूज आ रहा है, तो आपने बोला है बज वर्ड है, तो डेल्टा क्रॉन इज ओनली अ बज वर्ड या अदर देन ओमिक्रॉन? क्या है डेल्टा क्रॉन?
It has like a reported from the Cyprus, okay, I think couple of days before, and which I have already tweeted that we don't need to worry about it because first of all, it is a natural phenomena where the co-infection can happen, number one. But this Delta Cron you were talking about, it was just like an error in the lab where some samples got mixed up, okay, and because of that, they were getting the signal for the Delta also as well as for the Omicron also in their study, which has been like highlighted day before yesterday by CNBC and other channels also. And the lab also has agreed that yes, there has been some like a mixing up of the samples. So there is no Delta Cron as such.
In case of Omicron, what will be the isolation period? Earlier it was the 14 days. Nowadays,
I think Dr. Saab has already stated कि पहले 14 दिन था, 10 दिन कर दिया, 7 दिन कर दिया है. Dr. Saab has already responded to that. I think Mr. Khan, please carry on with your word of thanks, please.
अच्छा, I hope I am third time lucky.
Please carry on, sir. Please carry on.
So Dr. Saab, thank you very much for a very elaborate narrative. So we've been hearing a lot about these, there've been a lot of deliveries and lectures, but then there was something a little different in yours. Not only because you spoke in English and then in Hindi also, that made it very natural for us. And the good thing is that you're also supporting the fact that Omicron may become an endemic. So it should be the end of our miseries. And also you have given clarity on the booster doses, which was so far missing. That is, you have made it a little more flexible. I mean, I don't think we have to really go for 9 months. If the world opinion is 4 to 6 months, that should be good enough. And that we have only, you rightly said, the beggars are not choosers. We have only option of 2 vaccines at the moment. So I think we'll have to go for whatever is best is available, whatever our doctor says.
True.
And so thank you very much. I think you said so much and everything is very clear, crystal clear. Thank you, Rajiv ji. Thank you, Gaurav. And thank you, Mathur Saab, for bringing this session on. So thank you very much. We close the session now.
Yeah.
Thank you.
Thank you very much, Dr. Saab, on behalf of LMA and the YMF also. And I'll request everyone to take care and stay safe. Thank you very much, Dr. Saab.
Thanks a lot. Thank you very much to all of you.
Thank you.
Thanks
Thank
a
you.
lot. Thanks a lot.
Yes, ठीक है.
Okay, thank you.